Hello, good morning, everybody. My name is Laura Fernandez. I'm product manager here in Condalab. And the first thing I want to say is thank you all for coming, for joining this webinar today. So just a little brief reminder for those in case that you still don't know who Condalab is. I'm gonna show you. So Condalab, we certainly started like more than 60 years ago. We were agarose manufacturers. Also, we produce microbiology, culture media, and bioingredients. But then something like 20 years ago, we moved into another application in other areas, and we started in the molecular biology part with cloning-related products, PCR, nucleic acid preservation, but also some other areas like epigenetics, genome editing, of course, NGS. But then we added to our portfolio the cell biology part with viral transduction, cell function, cells and tissue models, or even cell immortalization. But please do not forget that we also have some equipment related to nucleic acid isolation or PCR. And we also can provide services like NGS or, as I said, gene editing. So let me share. Okay. Yeah, I'm switching off my camera so I can introduce to our speaker today. We have today our specialized partner for oligo improved production, who is Metabion in this case, and they will let us know about the very promising work of therapeutic oligonucleotides. But before we begin, I'd like to remind everyone that, of course, you can use the chat box to ask any question, and we'll cover them all at the end of the presentation. Our speaker today is Dr. Marike Hansma, who obtained her BSc in Pharmaceutical Science and MSc in Molecular Biology from the University of Groningen in the Netherlands. She did her PhD in Structural Biology at the Ludwig Maximilians University in Munich, where she focused on the DNA repair proteins, and then she joined Metabion this year as a product marketing manager. So good morning, Marike, and thank you for coming. You're in control right now, so let's start with the therapeutic oligonucleotides.
Good morning, everyone, from my side as well, and thank you, Laura, for your kind introduction. So my name is Marijke, and I recently joined Metabion as a product marketing manager. But based on my scientific career, I already have a lot of experience with oligos. Actually, my PhD in Munich, I was also a Metabion customer, so it's for me also nice to continue in this field. Yes, so and today I'm going to talk to you about unlocking the potential of therapeutic oligonucleotides, and I'm looking forward to take you with me on this little journey this morning and explain you about this very exciting field. First, I want to talk a little bit about Metabion. So who are we? So we are a small company in the Munich area, and we specialize in producing custom high-quality oligonucleotides. And we actually have the most complete portfolio of any modifications. Think of fluorophores and quenchers and all kinds of other chemical modifications, which you could use to make your own very customized oligonucleotide for whatever purpose you need it for. So the company has been around for quite a while. We were founded in 1997 by Dr. Regina Bichelmeier, who is still our CEO today. And as I said, we're based in the Munich area. It's very nice here in the south of Germany, close to the mountains. We have our HQ in Planegg, which is a suburb of Munich, and we have a very new state-of-the-art production facility in Kirchheim, which is on the other end of the city. And we are ISO 9001 and ISO 13485 certified, and we have also GMP, GDP-like production capabilities, for also, for example, therapeutic approaches. And we have a worldwide customer base in more than 80 countries. So that is about Metabion. Now I want to take you through the agenda of this presentation for today. So first we're going to talk a little bit about the classification of therapeutic oligonucleotides. Then we are going to look at the oligonucleotide therapeutic market, what is going on there. We will have a little look at the mechanisms of action, so how do these drugs actually work. Then we're going to look at the chemical modifications, delivery strategies, And also we're going to look at some real life examples, so which oligotherapeutics are already on the market and are approved. And at the end, I also want to talk a bit about meta-beyond solutions for therapeutic oligonucleotides. So what are therapeutic oligonucleotides at all? So basically, these oligonucleotide therapies that typically short, single or double-stranded RNA or DNA molecules that can be used to treat a wide range of diseases. And very often these are very rare genetic diseases for which there's an unmet need. Typically, they bind via Watson and Crick base pairing to enhance or to repress the expression of target RNA. There's one exception to this Watson-Crick base pairing. We'll come back to that later. They're often very heavily modified to increase stability and tissue targeting because DNA and RNA are relatively vulnerable molecules, right? So they need to be modified. They're very rapidly expanding category of drugs, and they also will change the standard of care for many diseases, and they will also enable personalized medicine on the longer run. So here's an overview of the different types of therapeutic oligonucleotides. So one of the most famous classes are the antisense oligonucleotides, or the ASOs, which are single-stranded molecules. They modulate the gene expression, for example, in genetic disorders, cancer, and viral infections. And another well-known class of therapeutic oligonucleotides is the small interfering RNA, or the siRNA, and they use RNA interference to silence genes, also a way to treat genetic disorders or cancer, for example. MicroRNAs are also a class, and they regulate translation, and they also find applications in treating cancer, cardiovascular disease, or neurodegenerative diseases. Then here's the aptamer. And aptamers are slightly unusual because they are actually not binding through Watson-Crick base pairing to other oligos or DNA or RNA. They're basically also binding to proteins, yeah? So they can be really used for selective binding for targeted drug delivery, for example, by really targeting the molecule to a specific, say, protein or cell. Also used in cancer treatment, infectious and autoimmune diseases. Then we see here the single guide RNA. This is for CRISPR-Cas9 gene editing, which is all very well known, of course. And another class are the CpG oligos. Now, these are not therapeutics in their own right. They're adjuvants, so they're usually added to, say, vaccines or to immunotherapies to trigger a stronger immune response. And basically, these are also short, single-stranded oligos, which have a CG motif. So that's where the name CpG oligo comes from. So these are the different types of therapeutic oligonucleotides out there. And over 20 nucleic acid therapeutics have been approved in the past 25 years. And we see here a little overview. Moment, I just look for my, oh, no, I'm zooming in, sorry. I was looking for my laser pointer. I'm sorry for this. So over 20 have been approved in the last 25 years. So the first one was Fomifizan, which was approved in 1998. And you see that over the years, more and more of these oligonucleotide therapies have been approved. So it's really exponential growth. And actually the market is also predicted to grow by over $10 billion in the next 10 years, yeah, because there's so many new drugs coming onto the market. The majority of them treat orphan or rare diseases for which there's no other treatment available at all. So what are the most common types of oligonucleotide therapeutics? We see here this pie chart, and you see the biggest slice of the pie in blue. These are the ASOs. And in orange, you see the second biggest class are the siRNAs. So these are the vast majority of the oligonucleotides that are currently on the market and also under development. And we see here another pie chart, and I just want to show here quickly for which diseases they're mainly used. So the largest category are metabolic diseases. They use oncology, neurology also to hold a lot of promise there, ophthalmology, and also for gastrointestinal diseases. So these are the main classes of diseases for which these oligos can be used. So in terms of market share of therapeutic oligos, so this is an overview of some approvals by modality from the FDA in 2023. And you can still see that the majority of the new approved drugs are still classic small molecule drugs. You can also see here that in pale orange that proteins are also a very big class, so in particular monoclonal antibodies. But you can also see that in the pie, there's also now a part that is for the oligonucleotides. And as we saw already before, mainly antisense oligonucleotides and also siRNA. But what are the challenges for therapeutic oligonucleotides? I mean, RNA in particular and also DNA are very vulnerable molecules. So what are challenges for these therapeutics? So one of the biggest issues is, of course, target affinity. You want the therapeutic oligo to interfere with the right gene, right? You don't want to bind it somewhere off target because that can cause all kinds of side effects and also makes the therapy less effective. So target affinity is a challenge. Another problem is side effects and toxicity. Of course, if the oligo binds to, say, the wrong gene, to the wrong RNA, silences the wrong gene, that can cause severe side effects. And also RNA and DNA can be very immunostimulating. So they can also send the immune system in overdrive and also cause toxicity through this. So this is also one thing that is an issue. And as I already mentioned, they are very vulnerable molecules. So metabolic stability is also simply an issue because RNases are everywhere, nucleases. And these RNases are basically one of the first lines of defense our body has against viruses, for example, which often contain RNA, obviously. So RNases are everywhere to degrade the RNA the moment it's encountered. So that's also a major, major challenge for therapeutic oligos. Pharmacokinetics and pharmacodynamics also not so easy. The body typically gets rid of oligos in the bloodstream really quickly, typically through the kidneys. So they have a very short survival time in the body, on top of the RNases that are also there. So pharmacokinetics, pharmacodynamics are a challenge. And then lastly, delivery is also a problem. You want, of course, that the oligo goes to the part of the body that is affected by the disease. I mean, for example, the brain or the stomach. And that's why also targeted delivery is like quite challenging because typically the body does its best to get rid of the RNA, but we have to get it also to specific body parts. So how do we do that? And one of the main solutions for these challenges is chemical modifications. By chemically modifying your oligo, you can increase the target affinity, you can reduce the side effects and toxicity by giving the oligo more drug-like properties. You can make the oligo also more stable and confer stability against RNases, for example, so they cannot be chopped up by the enzyme. Also using chemical modification, we can improve the pharmacokinetics and pharmacodynamics. And we can also use chemical modifications to really target the oligo to a specific tissue or to a specific cell. And I want to show you a few examples of this. So here's the first challenge, stability. So we see here DNA or RNA molecule, this chemical structure. So what can we actually do in terms of chemistry to really increase the stability? So I like to zoom in a little bit here. So one of the most popular solutions, which you also see, we will see in practice also in a few practical examples in a couple of minutes, is by introducing phosphorothioate modifications, where you replace this little oxygen molecule by a sulfur atom. And this really conveys a lot of resistance against nucleases. The nucleases can simply not chop such a molecule that has this phosphorothioate as easy as, say, the normal backbone. Yeah, so this is really helpful to help against nucleases. Then you can also make changes on the level of the ribose. This is the yellow part here. A very popular option is to create the so-called locked nucleic acid. So you introduce an additional carbon atom in this ring and basically, to stabilize the system basically. And this also increases the specificity and sensitivity of the oligo quite significantly and also helps a bit against nucleases as well. To have like this locked nucleic acid modification. So that's one thing one can do. One can also make sugar substitutions. So this is basically, you can introduce, for example, a fluoroatom or methyl group to also basically stabilize and enhance the binding affinity of the oligo. And furthermore, we can also change nucleobases here in blue. So, and the advantage of doing that is that you reduce basically immunogenicity and also you improve the specificity by changing nucleobase modifications because the body does not recognize it anymore so much as something dangerous somehow. I mean, the immune system doesn't recognize it as well anymore as some kind of dangerous DNA, RNA that needs to be degraded. So that really helps to improve the reduce the immunogenicity. Yeah. and specificity. So this way you can play around with a lot of options to really stabilize your oligo so it survives longer in the body and binds better to its target. And then the other challenge is to deliver to the right organ and into the cell. And their bioconjugation helps really a lot for tissue-specific delivery. So I show you now a few popular options, and I think one of the most famous one is by introducing an N-acetyl galactosamine moiety, which also has the shorter name GalNAc. And GalNAc really helps to target the oligo towards the liver. Typically, it's conjugated in a more complicated form than just this basically the simple sugar. Typically, it's this trivalent molecule with this long tails. So this is like this trivalent GalNAc that's then conjugated to the oligo that really targets it to the liver. I will show you an example of this later on. But first, I still want to show you a few other options. One can, of course, also couple a peptide to your oligo. In this case, I have an ASO as an example. And this can, for example, help to target a specific cell and also help the oligo to really go into the cell. The oligos, you have to keep in mind, are very negatively charged. And it's for them, because of the negative charge, very difficult to penetrate the membrane. But with a peptide, you can enhance this cell permeating abilities, so to say, of this oligonucleotide. So that's a very elegant method. Another very nice approach is to couple a lipid or some molecule, like in this instance, cholesterol, to your oligo. And interestingly, you can use this very much to target the brain. The brain is actually a very fatty organ, so having a lipid attached to your siRNA or to your ASO really helps to target the oligo towards this body part, towards for brain diseases. This is very promising. Then you can, of course, also use antibodies. I mean, antibodies you can basically engineer and make them bind to anything. Yeah, so you can make antibody, siRNA or ASO conjugates to target them to also specific tissue, specific cell, the specific protein. And lastly, you can also, of course, use an aptamer to couple this to another oligo. So basically, you have one very big oligo in which the aptamer part really binds to a specific target, and then the siRNA or the ASO does its work. Yeah, so I think this is a very elegant solution as well. Now, I want to delve a little bit deeper in GalNAc because this is such a popular option for delivery to hepatocytes. I just want to show you quickly the mechanism of how such a thing then works in practice. So we have here our liver, and basically we see here, very simplified, a hepatocyte, and on this hepatocyte there is this ASGR receptor, and this ASGR receptor, this recognizes GalNAc. So we see here now a GalNAc modified oligonucleotide, and basically when this is binding to this ASGR receptor, the cell takes this whole complex inside. So this is ASGR mediated uptake. It is internalized by the cell, goes into the endosome, and before the endosome is then merging with a lysosome in which everything would be degraded, the oligo can escape from the endosome, and once it's out in the cytoplasm, it can do its function either through mRNA degradation or gene silencing. I will come to speak about this in a minute in more detail. But this is essentially how GalNAc works. And there's already several examples of oligos with GalNAc on the market. Donzera, for example, which was very recently approved, and also inclisiran. These have this GalNAc moiety. I will explain them in a bit more detail later on. So here's the overview of the different types of oligonucleic acid therapeutics once again. And I now want to focus a little bit more on two specific ones, the most common ones, the ASOs and the siRNAs, and really go a bit into more detail on how they work mechanistically and discuss a few examples with you. So here's the ASO. Antisense oligonucleotides, so just as a reminder, these are short single-stranded nucleic acids that bind to specific RNAs to modulate the gene expression. And they are used to treat genetic disorders, cancer, and viral infections. So what's the mode of action? Interestingly, there's actually three modes of action, but they all start basically by binding to the mRNA target. So we have here our target mRNA, and then one of the three following things can happen. The most common mechanism is RNA cleavage through the recruitment of RNase H. So of course, this having a double-stranded RNA in the cell, that is of course something that is suspicious. That could be a virus. So the cell has mechanisms to get rid of this double-stranded RNA molecules, and one of these mechanisms is indeed the recruitment of RNase H. And what RNase H does is it has endonuclease activity, means it cleaves the target at variable sites, and then all these products are released basically from the RNase H, so cleavage products, and also our oligo or therapeutic oligo. And basically the consequences, of course, when the mRNA is cleaved, the protein cannot be formed anymore. That is one mechanism of action. You basically just destroy the mRNA that codes for the problematic protein. Second option is also steric hindrance of the ribosome. So of course, a double-stranded mRNA cannot be loaded in the ribosome. Yeah, it can only basically translate single-stranded RNA, obviously. So basically, the ribosome gets blocked, and this way also translation cannot take place anymore, and the protein is not produced. And thirdly, splicing modulation is another mechanism of action. So basically, you can also use these ASOs to induce exon skipping or inclusion. And this way, you get a different variant of the protein, so differently spliced variant that either includes a certain domain or a certain part of the protein or leaves it out. Yeah. So this is also playing a role in certain diseases, such as Duchenne muscular dystrophy, for example, where also the protein is there, but it's just not the correct variant of the protein. So this is basically the three different modes of action through which ASOs do their job. And now I want to show a few examples of approved ASOs. So the first one I want to highlight is vitravine or fomivirsen, which was the very first therapeutic oligonucleotide. It was approved in 1998, and it was actually created or developed to treat cytomegalovirus-induced retinitis, so an infection of the eye. And basically, this is a disease that was very common in patients suffering from AIDS or HIV, because they have no immune system anymore, and they're very, very susceptible to viruses that for a healthy person would not be a problem. And one of the most common diseases, or fairly common, was this type of eye infection, which is caused by the cytomegalovirus. So what vitravine targeted is basically a viral protein, the CMV-EIE2. Yeah, and basically it makes sure that this protein is degraded. And what vitravine also did, it was stimulating the immune system through toll-like receptor 9. Yeah. And it was a 21-mer ASO, so it's a relatively short molecule, and it's a full PTO, so it has everywhere these sulfur molecules introduced, so the RNases cannot cleave the backbone anymore. And in this case, it was actually a DNA molecule, so it was very resistant against nucleases. Now, actually, vitravine is not on the market anymore because thankfully we have very good medication nowadays to treat people with HIV. So basically, there's very few people who suffer from this disease nowadays. So this vitravine was taken off the market because there was no need for it anymore. So a more recent example is Donzera or donidalorsen, which was approved actually last month by the FDA, so in August 2025. And this is the most recent ASO, and it's used to treat a disease called hereditary angioedema. And this is a disease in which the patients randomly get terrible swelling of, say, the hands and their feet, and it starts kind of spontaneously. It's a very, very problematic, very terrible disorder. And the problem in this disease is that in the liver, there's a misregulation of a protein or overexpression of a protein called prekallikrein, which through a complicated pathway, which also involves another protein called bradykinin, regulates the blood pressure. And when this system goes haywire, that can lead to this terrible disease. So what Donzera basically does, it targets this prekallikrein expression by also activating RNase H1. And you can see here chemically what happened in all 30 years of science, right? It's a much more complicated molecule. It's a so-called gapmer. So it has basically, say, this DNA core, so to say, and it has different modifications on its ends. The first 5 nucleotides are also modified, and it has also this it's a full PTO again. So it has everywhere these little sulfur atoms introduced, so it cannot be attacked by nucleases. And this is also a protein that has this GalNAc moiety. And it's indeed also used to target it to the liver, right? So that all makes sense. There's another ASO that is Qalsody or tofersen, which was approved in 2023. It's a first-in-class drug, and it's used to treat familial and myotrophic lateral sclerosis. This is also known as motor neuron disease. So it's a rare neurodegenerative disorder in which patients get completely paralyzed, and there's no treatment for this disease whatsoever. Nothing you can do. And this is really the first treatment. And in these patients that suffer from this disease, they have a mutated form of an enzyme called superoxide dismutase 1 or SOD1 that basically clumps together in the neurons. It forms aggregates and that causes the neurons to die. And this way, these patients get paralyzed. So basically, what this Qalsody does is that it degrades this protein through RNase H1 activity. So this protein that's aggregating is then not there anymore. It's also, again, it's a very short molecule, 20-mer ASO, and it has basically RNA, DNA, RNA, gapmer structure. And in this case, you can see it has a mixed backbone. So it has not modifications everywhere. It has them just in a few spots. So there's like different variants you can see of this type of oligo. I want to change gears a little bit and talk about another class of molecules, namely the small interfering RNAs or the siRNAs. And just as a reminder, these are short double-stranded RNA sequences that degrade specific mRNAs to prevent protein translation. And this RNA interference mechanism is then used to silence genes linked to cancer and genetic disorders. So what's the mode of action? So RNA interference is directed by exogenously delivered siRNA. So we have here this siRNA that, despite its very negative charge, enters the cell. Yeah, and then it loads a protein complex called RISC. The RISC stands for RNA-induced silencing complex. So this siRNA is recognized by the RISC complex. And what the RISC complex then basically does, it removes the so-called passenger strand from the complex and then uses the guide strand as a template. And this is now very interesting because this template, this is then what recognizes the target mRNA, which we want to get rid of, the mRNA that codes for example, a problematic protein. And what then the RISC complex does, it will cleave this target mRNA, and then the products are released, and then the catalytic mechanism is repeated. Yeah, and then more and more of this mRNA is degraded, and this way the gene is basically silenced and the protein is not expressed anymore. So that is essentially how RNA interference works in a nutshell. And I want to show you a few examples of these small interfering RNAs that are currently on the market. So one of the first ones that was Onpattro or patisiran, which was approved in 2018. It was the first therapeutic siRNA and is used to treat hereditary ATTR. And this is a disease in which there's amyloidosis in the neurons, basically. So basically in amyloidosis, there is also it's a disease in which proteins aggregate and form clumps, in this case in neurons. So this causes neuropathy. And basically this Onpattro is targeting, again, the liver. Yeah, and it reduces the protein that is causing this disease. It's called transthyretin. Sorry if I pronounce it wrong. And this is the protein that really forms these clumps, these amyloids in the neurons. So by targeting this protein, you can treat this disease. And you can see this is also chemically a bit more of a complicated structure. So you see it has these overhangs and it has a lot of modifications, different types of nucleotide modifications internally to stabilize it and to target it to the right mRNA. And interestingly, this one has also slightly different delivery system. You might think, like, why does it not have this GalNAc modification if it's targeting the liver? In this case, a different delivery system was used. This is typically packaged as a lipid-based nanoparticle. So you can also use this type of carrier to deliver, say, an oligonucleotide to the right cell, to the right organ. So another drug that was also more recently approved is inclisiran. And this was also first in class, and is one of the first for cardiovascular disease, so the first oligonucleotide for one of the first for cardiovascular diseases. And it's mainly used to treat heterozygous familial hypercholesteremia or clinical atherosclerotic cardiovascular disease. So in these patients, they have too much cholesterol in the blood because the metabolism is not working properly, and they're very prone to heart disease because of this. Um, oh, and basically, it's targeting the liver, protein in the liver, lowering the LDL levels. And you can also see, again, this chemical structure is quite complex. It has, again, also some of these PTO modifications to stabilize the backbone, and it has this GalNAc moiety as well. So with that, I would like to change gears a little bit. Now we've talked a little bit about oligonucleotides and we've seen a few examples. Now I would like to talk a bit more about what MetaBeyond can specifically offer in terms of therapeutic oligonucleotides. So our expertise, so we have almost 3 decades of experience in manufacturing high quality custom oligonucleotides. Yeah, and we can really offer very comprehensive and tailored solutions from the very start in the research and discovery phase to also non-GMP and GMP-like contract manufacturing for preclinical studies. Yeah, we have extensive production capacities for timely project realization, and especially in the early stages and also the beginning of scale up for the initial preclinical studies. And we have a dedicated expert team for therapeutic projects. And our portfolio covers all those different types of therapeutic oligonucleotides we've already seen early in the presentation. So we can make ASOs, we can make siRNA, miRNA, aptamers, single guide RNA, and also the CpG oligos. And we can also make all kinds of modifications to increase the stability and specificity. We can introduce also fluorophores for like the very initial studies to see how, for example, the oligo is behaving in the cell. You name it, we can probably make it. We can also make all kinds of conjugates for this tissue-specific delivery. We have an exhaustive panel of QC analytical methods and services, so which we typically apply on the raw materials and also on the final products to meet your specifications, which include also mass spec to check for identity. We can assess the purity by UPLC and HPLC. And of course, we can check the appearance, pH, osmolarity, quantify the bioburden and endotoxins. Especially endotoxins bioburden is very, very important before you really go into animal tests and patients eventually, right? You want to make sure there's no endotoxins in your oligo anymore, which can make the person very sick. Also, we can test for residual solvents, heavy metals, water content, sodium content, and all those things you need to know before you proceed with your preclinical studies, right? To make sure you have good quality material. And we also offer additionally custom formulation, fill and finish support and services. So we have very extensive downstream processing equipment and capabilities. So we have chromatography systems, ultrafiltration equipment, lyophilization services, liquid and powder filling, and we can also do stability studies on request to see really how long-lived the oligo is. So with this, I already come to the end of my talk. I just want to give you one more quick summary, a take-home message. So first about therapeutic oligonucleotides. They are a very highly promising new class of drugs for unmet medical needs. Stability, targeting are still challenges, but there has been significant progress thanks to chemical modifications and also smart delivery systems. This also solving most of the problems we have with stability and targeting. So there's a lot of potential in the next years. In terms of MetaBeyond, so we offer 3 decades of experience in high-quality custom oligosynthesis. Our portfolio covers all types of oligo-based therapeutics, modifications, conjugates, which you would need for your studies. And we have comprehensive custom solutions from start till, say, the GMP-like contract manufacturing for preclinical studies. From start to, say, the end of this, we can cover you. And we have state-of-the-art facility equipment equipped with cutting-edge devices and technologies to make this all possible. And of course, also a team of great scientists. So here are a few references also for further reading. There's a lot of very interesting papers out there, which I can highly recommend. And I think this is a good start to look at a few of these reviews and papers. some of the images where they come from. And with this, I would like to thank you very much for your attention, and I'm open for questions.
Okay, thanks, Marie. That was really a fantastic overview of the therapeutic oligonucleotides and certainly what we could expect from them and the current challenges. So let's see if we have any questions in the chat box. Okay, I have one. Yeah, first of all, many thanks for the excellent clear talk. Also from my side, Marie. I was wondering if it would be possible to have a PDF copy of the presentation.
Yes, of course. Yes, we can make it available to you.
Yeah, as we will have the contact, we will send it to you. We will get it, the or the talk in PDF, and we will send it to you. No problem. Okay, so let's see if we have any other question. Okay, yeah, there is one more. Okay. Okay, yeah, okay. I will send to that email address. Perfect. So we have some other questions. What diseases can we target next?
Yeah, so I think there's many diseases you can target with these oligos. I think it holds a lot of potential for cancer treatment, but also for neurodegenerative diseases. For cancer, there are already a lot of therapies, but for example, for neurodegenerative diseases, there's not much we can do for most of them. And I think as these are often caused by also protein aggregations, for example, I think there's a lot of potential targeting these proteins that cause the aggregation with this type of therapeutics. That would be one example. And I think also in general, I think there's a lot of potential for personalized medicine with these therapeutics because, I mean, oligos in principle chemically relatively easy to make. And you could maybe also quite easily adjust them to a specific patient. Yeah. At the moment, it's still a bit tricky, but I think in a few years it will definitely be possible.
Okay. Yeah, we have another one. How do we know that the therapies are really working?
Yeah, of course, you have to really do a lot of tests, right, to figure out. But there's different things you can do. You can, for example, really measure, say, the target engagement. For example, with qPCR to really see, like, are they actually interacting with the target? Is the oligo really binding? So that would be one thing. And the other thing is also to look at the readout. Is the protein I want to get rid of really less prevalent in the cell? So this is also what you can measure. And you can, of course, also look at biomarkers in the blood.
Okay, thank you, Marie, for your answer. We have a new one. Can I ask your thoughts on using PNAs or mimic DNA as oligotherapeutics?
Excuse me, can you
Yeah.
please repeat it? Which one?
Can I ask your thoughts on using PNA or mimic DNA as oligotherapeutics?
PNA. This I would actually have to check. So PNA at the moment does not really ring a bell. Sorry.
Can you please, Salvatore, just type in PNA?
PNA. PNA.
Let me see.
Well, what does it stand for? Maybe that rings a bell. Sometimes there's so many of these, like this short version. So sometimes I forget like what is.
Peptide nucleic acid.
A peptide nucleic acid. Ah, okay, thank you. Yeah, I discussed this already. Peptide nucleic acid.
Thank you.
Yeah, I think they can also confer more stability, for example. So I think there's also different potential for those. Yeah.
Okay. There's a new question. What is the regulatory about? Are you helping with that issue, let's say?
With regulatory.
Yeah.
So we do offer a lot of support for also customers who are interested in therapeutic oligos. So we are at the moment not really supporting clinical studies. We do more the preclinical, but I can already reveal that we will have soon a collaboration with a larger CDMO actually, where we can then really help your projects from beginning, from the initial R&D to preclinical, which MetaBiome can already cover. And through this collaboration, we can actually also cover then the clinical studies. yeah, it's not officially released. Will be very, very soon. So please stay tuned and look at our LinkedIn page and our website. Then we can say a bit more about that, because then we will also be able to support clinical studies in the future
Great.
as well. Well,
That's good
yeah,
news. Certainly.
very exciting, very exciting. Yeah.
And okay, we have a new one. So, what's your view of the future of therapeutic oligonucleotides when you compare it to other advanced therapies, such as those based on mRNA or even CRISPR?
Yeah, I think there will be a whole portfolio of different drugs to treat different diseases, right? I think that there will never be the case that one drug can really cure every disease. It would be nice if there was something like that. But I think it will always have its niche. So I don't believe that, for example, CRISPR will replace oligonucleotide therapies like the ASOs or the siRNAs, because they have a different niche. Yeah, you can treat different types of diseases. I think CRISPR is still I think for treatment still in its relatively early days, whereas ASOs and siRNAs have been around now for a while. They're also quite, I would say, relatively easy to make. Yeah. So I think there's a lot of future for those. Yeah.
Okay. So I think that maybe we can leave it here, but just in case if any other citizens has a new question, you can contact us and for sure we'll answer it.
Mm-hmm.
But I have to remind you that once that we're finished, you'll receive like a short survey. So please complete it so we can improve in it. It will only take I promise, just a couple of minutes, and it will be really helpful for us. And we make to the end of this journey. Hope that you have enjoyed as much as I've done. And you all have access to the recorded version too, so you can rewatch it or share with your colleagues. And please, of course, as I say, do not hesitate to contact us if you have any further questions. Thank you all for attending. Thank you a lot, Marike, for this amazing webinar, I have to say. And I
Thank you so
wish
much.
you all a great week.
Thank you, Laura, also for having me. It was really nice to be here with you today.
You're welcome. Okay, so have a great day, Joe.
Yeah, have a good day. Bye-bye.
Bye.